Hello everyone, my name is Dawson Payne, and today I'll be presenting our abstract, which is titled An Updated Genome-Scale Metabolic Network Reconstruction of Pseudomonas aeruginosa, PA14, to characterize mucin-driven shifts in metabolism. So first off, I want to give you guys some background on Pseudomonas aeruginosa, and this is a bacterial pathogen that commonly infects humans, especially in the context of cystic fibrosis. And it's typically treated with antibiotics. However, as the use of antibiotics has been on the rise, we've seen more and more multi-drug resistant strains of Pseudomonas emerge, and these are causing serious problems. Over here on the right, I have some pictures of Pseudomonas and a bunch of different phenotypes that it's capable of producing, which just shows how rich its metabolism is, which makes it really interesting to study. So now, to help us understand the metabolism of Pseudomonas aeruginosa, we produced this, what we call a genre, which stands for a genome-scale network reconstruction. And this is basically a mathematical model of the metabolic capabilities of an organism. So kind of quickly, the way that we construct these is we start at the genome, and from the genome, you look at the genes that it has, the proteins that those code for, and then the reactions that those proteins facilitate. So here we have some example stoichiometry. You can then take all those reactions and store it in this giant matrix, and you can kind of construct these models that we build to make them more biologically relevant. So you can see here we have like what would be a bacterial cell, you have things coming in, it can do these metabolic transformations, and then also kick things out and grow. So now on to IPAL 1129, which served as a starting point for this updated reconstruction. And this was actually a model that was produced in 2017 by Jennifer Bartel and Anna Blazer, who are alumni of my lab. And it's an excellent model, and they originally used it to give insights into like growth virulence trade-offs. And this is a really well-produced model. Over here on the side, we have kind of the different kinds of metabolism that it accounted for. But as we've been gathering more and more knowledge about Pseudomonas and gaining more insights, it's important that we update this model to make it more accurate and help make better predictions. So some key areas of improvement that we worked on was improving prediction accuracy. So there are some kind of key predictions that it was getting wrong that we reconciled with the experimental data. We expanded the metabolic coverage. So this was done by adding different genes and reactions and metabolites to the model. We resolved this energy generating loop, which is allowing the model to kind of generate ATP for free, which is doing various bad things such as inflating growth rate predictions. We also updated the biomass formulation to include lipopolysaccharide, and we improved the annotation within the model, so this makes it more compatible with different databases and kind of easier for people to use. And we also upgraded the formatting from SBML level 2 to level 3, which allows you to use kind of different packages and gives you different data structures within the model, which are helpful. So our updated reconstruction we named IPAL 21. And here's just a quick rundown of the different things within the model, such as genes, metabolites, reactions, and the number that our reconstruction contains, and then also compared to the previous version, how much we added. And this can be a little misleading because, for example, in the genes case, we actually added more than 40 genes, but we also got rid of some that didn't make much sense. So you're seeing the aggregate here, and it doesn't really tell the whole story, but it's helpful for understanding where our areas of emphasis were. Some things I wanted to point out was within this metabolite category, one thing that we did was we organized the biomass objective function into macromolecular categories. So now when you read it, instead of seeing kind of this jumble of a bunch of different metabolites, you're going to see things like RNA, protein, DNA, things like that kind of mean more biologically. Here on the paraplasmic metabolites, you'll see that there's only one, and this one was added by us. So we added this whole paraplasmic compartment, and part of that was to resolve that energy generating loop, which we were successfully able to do, which is great. But also just having the paraplasm there makes it more representative of the biology, which we thought was good. As well, within the reactions, kind of a major area of the metabolism that we expanded was the amino acid and glycophospholipid metabolism. So once we updated this model, we sought to do some validation. So first off, we took these growth rate predictions, which are shown down here. These gray bars represent experimental values of doubling times. And this is in the LB media. Here's the SCFN, which is synthetic cystic fibrosis medium. And then we have a glucose minimal media. LB stands for lysogeny broth, by the way. And then, so we have our experimental values in gray. Kind of in this gold here, we have the previous version of the model. And then here in blue, we have our updated version. And you can see that in all three of these different cases, our updated model performs better than the previous version. And then here in the glucose minimal media, we even get within that experimental range, which we're pretty happy about. Another form prediction that we used for validation was substrate utilization. So here we performed in vitro growth screens on all these different carbon sources to see whether or not Pseudomonas was able to grow on them. Of the 91, we saw that in 48 cases, Pseudomonas was able to grow, and the other 43 was not. And then we compared that to the growth predictions by our model. And you can see that overall, we end up with our 89% accuracy. We also compared our gene essentiality predictions to an experimental data set, and we got 91% accuracy. So now that we have an updated and validated model, we're interested in applying this model in order to provide some biological insights into how PA14 metabolism is modulated by mucus. So just to give a little bit of background, mucus is a slimy hydrated gel that lines all wet epithelia in the human body. So if you think of places like the lung, the oral cavity, and the eye, all these places are going to contain mucus. And kind of one of the functions of mucus is to act as a defensive mechanism, because you're also going to have microbes in those places. And it kind of serves to tame these microbes and keep them from doing harmful things to us. So the way we went about this was by integrating transcriptomic data, which was produced by Katerina Rebeck at MIT's lab, with our updated model using this Riptide algorithm, which was produced by Matt Jr., who was a postdoc in our lab. And the way Riptide works is basically you can take this transcriptomic data and use it as evidence. And then you can combine it with your metabolic reconstruction. And by putting those two together, you're able to get more accurate predictions of what is happening metabolically in the system. And the way the experiments are run is they had this kind of baseline condition where it's the ABT-GC media, and they supplemented that with mucins, which are kind of components of mucus. You can see here the MUC5AC is one type of mucin, and the MUC5B is another type. So they supplement with the MUC5AC, another condition with the MUC5B, and then the fourth condition, they supplement with glycans, which were isolated from the backbone of the MUC5AC mucin. And by doing all this, we end up with these 4 contextualized models, which we can then compare. So the first thing we wanted to do was look at how the active metabolism of these models was shifting. So here you can see our 4 conditions are outlined here in different colors, and we did an NMDS plot of flux samples from each of these contextualized models. So each point here represents a different kind of flux state of the model. And what we're interested in here is looking how far from our baseline condition and salmon here, all of these points are shifting. So you can see here, we have our baseline condition, and then the glycans, which were isolated from the backbone of the MUC5AC. Didn't shift all that much, but we see a bit. And then the MUC5AC, we see a pretty good shift here. And then the MUC5B is kind of the clearest shift that we see. So this told us that MUC5B displays the largest shift in active metabolism, which we found interesting. So in addition to active metabolism, the other thing we wanted to look at was kind of the network structure of these contextualized models. And we did that by comparing kind of the set of reactions that was not contained by all the models, which we called non-consensus reactions. So upon comparing those, you can see here we have each of the models going across in these rows. And then if the dots filled in, that means that that model compares a certain group of reactions. So you can see here, we have this group of 13 reactions that are shared by the MUC5B, the glycans, and the ABT-GC models, but not contained by the MUC5AC model. And this is interesting because that indicates that the MUC5AC displays the largest structural shift. And this is especially interesting when we compare it back to the active metabolism, because we see here that the MUC5B shows the largest shift in active metabolism, while the MUC5AC has the most different network structure. So that's cool, but we were more interested in the active metabolism case. So the next thing we sought to do was compare the MUC5B case to this baseline case and see actually how, why the metabolism is shifted, what pathways are being utilized more differently. And we did so by running random forest, and we're feeding in kind of all the flux values for reactions that were shared between these two models, and then looking at which ones best discriminated between the two. so the MUC5B and the baseline case. And here are our top 7 hits on the random forest, and we provided the reaction name over here, and then connected to the pathway that it belongs to, and then also showed whether it was being utilized more by the MUC5B or less by the MUC5B. And this was interesting because you can see here we have 2 hits in fumarate metabolism, and this was actually noted in the original paper that we pulled the transcript data from, so it was nice to see that we were recapitulating those results. And then we also got 5 hits in propionate metabolism, which is kind of a novel insight. And this is interesting because propionate is known to have beneficial effects on human health. It has anti-lipogenic properties, anti-inflammatory properties, and anti-carcinogenic properties. Another interesting thing is that it's also a byproduct of mucin fermenters, so other bacteria that can kind of ferment these mucins. It's a byproduct that they kick out as they break down the mucins. And this may indicate a cross-feeding mechanism between Pseudomonas and these other mucin fermenters. So when Pseudomonas comes into contact with these mucins, maybe it starts to ready its metabolism to consume propionate because it knows that mucin fermenters will be nearby producing those byproducts. And this is interesting because once validated, this could actually be used to produce antibiotics that target propionate metabolism. And this could be helpful in times where you have these multi-drug resistant strains of Pseudomonas and traditional antibiotics don't work. So this new kind of insight could be used to control those. So finally, some conclusions. What we did in this project was we generated an updated genre of PA14 metabolism with considerable improvements. We then validated the model through a variety of metrics, and once we're happy with the model, we combined it with transcript data to investigate mucin-driven shifts in metabolism, which yielded some interesting biological insights. In addition, I'd like to thank our funding sources. The NIH and the German Research Foundation provided funding for this. Here underlined, we have the co-authors on the project and the manuscript. And I'd especially like to thank Laura Dunphy for all that she taught me when I came into the lab. She's kind of my mentor. And then also Jason Papin for all of his guidance. And then here, we have other members and alumni of the lab. So thank you all.